Okay, thanks very much for the introduction, John. Thanks for everyone for coming along to listen. So I'm from Microsoft Research, which is obviously a company that makes software like Windows and Office and such like. But one of the roles of Microsoft Research, specifically in the context of Microsoft, is to look at new possible avenues for new businesses. And biology is obviously a rapidly growing area that is receiving a lot of attention from mathematicians, computer scientists, and so in a way, by looking at biology, we think there could be new businesses there. We're looking at biology through the context of trying to think about whether we can actually program biological behaviors, and that's one way that we see that Microsoft might be able to have an impact in this area. So I'm going to tell you a story today about some work that we've been doing. It's quite an academic story. It's a work in collaboration with University of Cambridge. So we're kind of motivated by kind of beautiful patterns that we see in nature, and a lot of researchers are trying to understand, how these arise. You get these really cool patterns, such as in the zebrafish. This isn't a real organism. This is just. But maybe one day we'll be able to create such a thing. But there's also quite amazing pattern formation in single cells as well. So actually, you have asymmetrical division that can initiate these developmental programs in cells. And so obviously trying to understand how this works is receiving a lot of attention. There's contributions from theorists, Alan Turing, one of them, seems relevant because this is a Turing gateway for mathematics. I just wanted to show this nice summary by Takeshi Kondo a number of years ago now. He was showing that you can get very simple patterns by just having what's called a morphogen, it's a signaling molecule. By reading off the gradient of the concentration of that molecule, you can get stripes such as this. If you've got two morphogens, you can get slightly more complex patterns. But if you have interactions, you can get even more complex patterns. And this is kind of the archetypal wave behavior that you see in a so-called Turing pattern. So Alan Turing came along and brought some theory to bear in terms of the formation of patterns in biological systems. And what I've just shown a little simulation here of the emergence of a Turing pattern, but the idea is that you have these two morphogens and they're able to interact with each other in an activation-inhibition relationship such as this, and that enables the formation of these rather remarkable-looking patterns. As a number of years passed, and eventually we found evidence of Turing's theory in a real biological system. This is perhaps my favorite example. This is the formation of digits in a hand. This is a scan of a real mouse hand, and on the right here we've got the development of the limb bud. And what you see is that you have a sort of a wave pattern of the concentration of something inside in this limb here. And if you perturb some of the genes involved in this network, then you actually alter the wavelength of this pattern here, and of course that leads to getting more fingers. And they followed up this work a couple of years later, and they actually showed that there's a little network here which involves two morphogens, Wnt and BMP, and interactions between them are mediated by its component SOX9. And then they showed that with certain parameter values relating to this network, then you would get the emergence of a Turing pattern, and then they compared that with some data. So this is sorry, that's just another summary of the same thing. So one of the things that is curious about this is that they've been able to figure out that a Turing patterning system is at play in a very complex system. This is a biological system with lots of different signaling pathways, lots of different components, and it's hugely complex. And so one of the things that we've been asking is, can we actually learn about theories of patterning outside of the context of all this complexity? Because it's very difficult to learn about systems like this. And so the approach that we're basically taking, sorry, same question, how can we understand biological patterning independent of these complications? Well, the approach that we're taking to this is to see the extent to which we can actually program biological systems and program pattern formation, such as a Turing pattern. If we can do that, then hopefully along the way we'll learn a lot about how biological systems are constructed and how they have chosen to produce the patterns that we see in nature. So we're asking this question at a number of levels. You can think about programming the behavior of molecules. So you might pick molecules that might interact with one another in a certain way. That enables us to build molecular networks, which might have a predefined behavior. Of course, those are going to be interacting with inside cells, so then you have to start thinking about interactions of cells, the biophysics of cells, which of course is going to be important if we're going to be able to create behaviors at the colony level. So we're a software company, we like making software, I like writing code, I like making software. So towards this activity we've been producing our own software tools which are freely available. You can go to our website and have a look at what we've been doing. But essentially this is done to help us to understand how to program these biological systems. So I'm going to tell a brief story about an example of programming a biological system, and it's along a trajectory to basically wanting to implement Turing patterns. If we can actually implement it with in a real biological system, then hopefully we'll have learned quite a lot about the organization of molecules and how that can lead to the formation of cellular patterns. This collaboration with Jim Hasselhoff's group. He's a professor at the Department of Plant Sciences here in Cambridge. A lot of the experimental work that you'll see is done by collaborator Paul Grant. I've done all the modeling work associated with this project. And so really, if we're going to be able to implement Turing patterns, we need a number of key components to get along the way. I'm not going to end this talk by showing you Turing patterns implemented in a real biological system. We haven't got that far yet, but we have made a big step, which is that we've been able to demonstrate independent cell-cell signaling channels in the context of a single cell type, which of course is going to be important if you're going to have two morphogens that are interacting. You need to be able to have two signaling pathways acting in the same cell that are going to be orthogonal to one another. Eventually we're going to need to thread those together in an activation-inhibition network, and we're going to need some kind of differential diffusion between these morphogens. These are conditions for Turing instability. Okay, so I'm going to focus on independent signaling channels. I'll give you a bit of a background about some of the components that we've used. So I should say as well, the target organism that we're using here is E. coli. So we're trying to engineer new genes into E. coli so that these bacterial cells will form Turing patterns. I forgot to say that. So we can take inspiration from existing efforts to engineer the behavior of E. coli. So people have shown that you can insert new genes that turn on a gene in an oscillatory way. So this is making an oscillator. That's GFP fluorescence being shown. There's examples of using molecular components to implement logic circuits. So you can think of doing AND logic over two signals, and the way this one will work is these M1 and M2 signals will sequester a repressor protein that inhibits transcription of a gene. And so when these signals are there, then YFP is able to accumulate, but only if both of them are there, because you've got two copies of this gene here. The one that's especially relevant to signaling is this communication circuit here, which introduces the component called AHL. AHL is a signaling molecule that bacteria use to communicate with each other to establish whether they're in a high density in a phenomenon called quorum sensing. And the way the molecule works is it can move inside and outside of cells, but once it's inside a cell, it can bind to what's called a receiver protein. And when it's bound to that receiver protein, it makes a complex that's capable of activating a downstream promoter called P-Lux, and there's other different promoters in different E. coli. What they did in this example, though, is they had that promoter connected to a coding sequence of a protein that will tell the cells to commit suicide. So it actually acts as like a negative feedback on the proliferation of these cells. So you can imagine that as the cells are growing in density, they're producing more and more AHL in the medium, and as that concentration increases, there's a higher chance that that will then go into a cell, bind to a receiver protein, and activate this gene, so that naturally gives a negative feedback that keeps the cell density down to a specific level. Another example that I find really, really cool, actually, is this example from Ling Chong Yu's lab. And what they did is they had two different cell types, and they engineered a communication between them that made these cells act in a predator-prey relationship. So the way that works is the predator synthesizes a signal that is sensed by the other cell type, the prey cell type. Once that goes into this cell, it binds to a receiver protein called LASR, which is then able to activate transcription of CCDB. That's the same killer protein that we saw before. So that's going to make the prey die So it's induced death from a predator. That's why they call it a predator. In return, the prey makes a different signal, which is sensed in the predator cell type, binds to a different receiver protein, which activates the synthesis of an antidote protein. And what this does is it stops the death protein from being active. So in a way, the prey kind of rescues the predator. It tells it to stop committing suicide, otherwise it would. And because we've got a predator-prey relationship, as we know from ecological theory, is that you can actually start to form oscillations in the abundance of those two cell types. So here we have a prey peaking and then going down to a trough. As the prey disappears, the predator is able to accumulate, and so on and so forth. So these are real cells with these new transcriptional programs inserted in them, and they're communicating with one another. So coming back to our first goal on the way towards implementing Turing patterns, can we actually take these two signaling channels and put them in the same cell and have them act independently? Well, to cut a long story short, no, you can't. It turns out there's significant crosstalk between these signaling channels, and so we use that as an opportunity to try and understand where that crosstalk was coming from and to see whether we could engineer out that crosstalk. And here's the demonstration of that. So just to see how much crosstalk there is, what we did is we took the LuxR component and the LASR component, we expressed those both in the same cell type, and then we probed the system with different concentrations of these two different signaling molecules. So C6 would normally bind to LuxR and activate this promoter here, but the problem that we had was that when we added C12 to these cells, we know that it's going to bind to LASR, but somewhere along the line we found that we were able to activate this promoter via this signal, and these are the data that show that. So it's not quite as sensitive to the C12 signal as it is to the C6. Nevertheless, we're getting a strong measurable response. There's two options. Either C12 is binding to the wrong receiver protein, which then ends up being capable of activating transcription, or it could be that C12 is binding to its cognate LASR receiver protein, but then that complex actually also could be binding to this promoter. So in a way, we've got like a chemical crosstalk, which is where the signal binds to the wrong receiver protein, or there's a genetic crosstalk, which enables this one to bind. And we characterize those two things as different. Obviously, they're different processes. So the question is, can we identify where the crosstalk's coming from? And to help us to do this, we built a mathematical model that described all the possible interactions in the system. So we have that C6 can bind to a LuxR, C6 might be able to bind to LASR, and then C12 might be able to bind to each of those as well. And then we might have genetic crosstalk here, or it could all be mediated via chemical crosstalk. So this reaction system describes all of those interactions there. And then we did some derivation to figure out what the equilibrium response to each signal type would be at that promoter. And what's nice about this equation here is it's parameterized by biologically meaningful quantities, such as KGS, which is this genetic crosstalk component up here, and then KGR, and then KR6, etc., etc. That's all embedded within this equation. So then the next job is to try and identify what the values of these parameters are, and that enables us to quantify the source of crosstalk in this system. So we did that by collecting a bunch of data, but specifically we devised a strategy that enabled us to change the levels of LuxR and LASR in the system. So you can imagine if you increase the amount of this, and of course we're going to get more signaling through this channel. If we increase the abundance of LASR, then we're going to increase the quantity going through this channel, for example. And so the way we did that is we used an inducible promoter called PBAD, which is turned on when you add this chemical called arabinose. And of course, the more arabinose you add, the more induction of the promoter you get, and so the more of the LuxR protein you would get in these cells. And what you see progressively in response to C6 is you increase the amount of LuxR in the system, we get a more and more sensitive response. This is kind of as you'd expect, because this promoter should be responding to increased levels of LuxR in the signal. And so you've got the measurements in the points here, and then the model is the solid lines and the regions. But what we also found is that C12 indeed also increased its sensitivity in response to increasing levels of LuxR. So that kind of demonstrates that a C12 LuxR interaction is functionally relevant in this system, demonstrating that KR12 is non-negligible, so that we definitely have some chemical crosstalk there. Then we did things the other way around, and we increased the concentration of LASR, and we observed a slightly different response this time. the response to C6 was actually unaffected by changing the levels of LASR, which meant that a C6-LASR interaction was not functionally relevant in these experiments. Whereas when we increased LASR and then measured response to C12, we did see an increase in sensitivity. Therefore, obviously this is going to happen, that's a cognate interaction, but demonstrably this interaction must be occurring as well. So through this experiments, we've demonstrated that there is a chemical crosstalk involving C12 and LUXR, but there's also a genetic crosstalk. And using parameter inference and modeling, we're able to actually quantify the amount of crosstalk in those two places. So since we had a genetic crosstalk, we wondered whether we could actually remove that crosstalk by changing the promoter sequence of P-Lux. So what we did is we looked at the sequence of P-Lux, and we basically proposed a number of different mutations to that sequence in the hope that we would be able to find variants that were either very, very LUXR dominated and very weakly dependent on LASR, and maybe even vice versa. So here are the mutations we looked at. This is just this very simple screen looking at two different concentrations of HSL and a negative control. And I'm going to draw your attention to two particular examples here. Promoter 76 was very interesting because we demonstrated got a very large response to C6 LUXR signaling, whilst having a very low response to C12 LASR signaling. So in a way, this is a candidate promoter that could be better than the wild-type P-Lux for mediating LUXR C6 signaling. The second one was even more surprising, that we were actually able to completely reverse the sensitivity of this promoter to C12 LASR and C6 LUXR. So we get a very strong response on this side and a very weak response on this side. And that's just by making just a very small change in the promoter sequence. And then we went back and we asked the model what the new parameters would be in this new promoter. And we found that indeed, when fitting to all the data that we had, that when we compare the wild-type promoter, that's a dark blue one, with our two new promoter variants, 76 and 81, this is the association constant for LUXR regulators for the promoter and LASR regulators for the promoter. And we see that the mutations do drop the sensitivity to LUXR, but more for the second one than the first one. But here we see the kind of quantification of the fact that we have actually increased the sensitivity to LASR in this promoter, in promoter 81, versus 76, which completely dropped. And this is a demonstration of those two promoters in response to different concentrations of signal. And as you can see very clearly now, we get a nice strong response to C6, very weak response to C12, and in the other promoter, a very strong response to C12 and very weak response to C6. So we've gone quite a long way towards optimizing, creating these new promoters that are independent of one another. And this is just some data showing what happens, again, as we control the levels of LUXR and LASR in the system. So I won't dwell on those. So now that we've made these two new promoters, the idea is we're going to put them inside the same cells and see whether they really are orthogonal in the context that eventually we want to use them in. So we express these two promoters in cells. We express the LUXR and the LASR receiver proteins. And we connected these promoters to two different fluorescent proteins so we could measure them in the same cell at the same time. The next thing that we did is we used the model to actually optimize the levels of LUXR and LASR, because as I showed you before on these slides, changing the levels of LUXR and LASR will obviously change the throughput through these signaling channels. So it was natural to ask, whether there was actually an optimal level that could be expressed at. And so using our parameterized model, we sought to optimize the functional response to C6 versus C12 in the first promoter, and simultaneously the fold change in response to C12 over C6 of the other promoter. So if we maximize this quantity, which will obviously functionally depend on LUXR and LASR, then we might be able to improve the amount of orthogonality in the system. And we found that there was actually an optimal combination of those parameters in terms of increasing LUXR and increasing LASR. This is relative to 1, which is the nominal expression rate that we had previously. And this is what it would look like if we reach the optimal combination. And so then in order to actually increase these levels, what we did is we changed the ribosome binding sites on these genes to increase the number of proteins that we produce per unit time. That enables us to increase the intracellular concentration. And so we came up with a number of different ribosome binding sites, and we express those all in cells, and we measure the response. So here we have an example of changing the nominal double receiver cells to a different ribosome binding site for LuxR, increasing the LuxR level, and along this axis, putting a different ribosome binding site on the LASR expression, increasing it this way. And then we had combinations of increasing them, and then at different combinations. And what we found is that this particular construct here with 2 modified RBSs was getting pretty close to the optimal combination now. And indeed, when we look at the data, there's really very good orthogonality between these channels. So this is the CFP response and the YFP response to C6 in the same cells, and this is the CFP and YFP response to C12 in those same cells. And as you can see, pretty orthogonal responses. So to test that this kind of works in a spatial context, and in order to really be able to parameterize, for example, maybe the diffusion rate of these molecules, we developed a spatial assay where we plated out our double receiver cells, and then we looked at them in response to sending sending signals C12 and C6 from different locations on the plate. So we have cells that just express C6, and just by virtue of diffusion, of course, they'll diffuse across the plate, and then the double receiver cells will sense that molecule at different concentrations depending on that diffusion gradient, and similarly for C12. And because we have those cells report turn on CFP when they see C6, down here we start to see CFP expression on the right-hand side of that plate. And because they express YFP in response to C12, we start to see YFP on the left-hand side of the plate, and these are the overlays. And so these are the different constructs that we had on the previous map. And indeed, the best one that we could see was this LuxR 33 LASR S175, which was this one here. This is what we considered to be close to the optimal combination. And indeed, it definitely gave the best responses to C6 and C12. It definitely reports C6, it definitely reports C12. Some of the other combinations you'll see didn't really turn on YFP in response to C12. Some of them didn't turn on CFP in response to C6. So can we use a spatial model to describe this behavior? If so, then we can parameterize that system, and then we can start making predictions about what would happen in slightly more complex arrangements of these cells. So a simple model, just modeling cell growth with a logistic equation. Then we have diffusion of the C6 and the C12. And then we have gene expression mediated by this transfer function that we parameterize from the other data. And so we can parameterize the cell growth by using a third fluorescent protein. Won't worry too much about that. But here we have an example of how well our model fits to the data for different of these different constructs. This is the favorite one, and these are some of the other variants. And each trace here, what you see is the different colors represent different distances from the signaling source. And the model is in the dashed lines, and the data is in the solid lines there. And you can see it does a pretty good job of reproducing those data. All we needed to do there was calibrate the diffusion rates. So that gives us a quantification of those diffusion rates as well. The last thing that we did was we established a more complex arrangement by putting different cells in a complex arrangement that had the following property. One of them would respond to C12 and turn on C6. The other one would respond to C6 and turn on C12. And then we sort of induce the system by putting both cell types in the middle. So what would happen is, one of them would see the C12, start making the C6, and then the other cell type would see the C6 that was being made and then start making the C12. So eventually they will sort of start turning on in the middle there, and then as they make more and more of the signal, it will diffuse out, it will move into neighboring areas, and then that will activate the same relay signaling in the other areas. And on the right hand side here we see what you saw in that video was the spread of that signal activating transcription and turning on the fluorescent protein. Okay. Might just show that again just so you can see it emerging. it's kind of interesting because it's very much an inducible system. If you don't put both cell types in the middle, you don't actually get this cascading effect happening at all. That's what you're seeing on the left there. So on the right, you can see it's starting to emerge now. Both cell types are creating their signals, and now the signals are beginning to spread out down the plate and turning them on in this arrangement here. So the question is, was that behavior be predicted by a model? Well, when we simulated the model with the parameter values that we'd inferred from all the previous experiments, we didn't actually get the right behavior, which is a bit of a shame. But we found that if we just increased the transcription rates of all the genes ever so slightly, then we would actually get the behavior that we saw. So this is an example of that simulation where we've actually increased the transcription rates. And you can see that qualitatively we are getting this inducible behavior over time, and in the uninduced case we don't get any signal production. And the reason that happens is because we're really near a bifurcation point. As we vary these transcription rates along here, we go from having a very uninduced state to having an induced state. So that's an example of where the model can give us some insight about how the system was really behaving. So I'm going to end there, just summarizing. So what I've shown you is a case study of actually engineering a system and using modeling to help optimize that system. Basically, we created these two mutant promoters, one of them that ended up being very much a LuxR signaling module, and another one being a different LASR signaling module, enabling independent responses to C6 and C12 signaling. And then using the model, we were able to actually optimize for orthogonal signaling by controlling the expression of these receiver proteins. And finally, we demonstrated that we have this independent signaling by creating these double reporter cells, and then showing that some of the spatiotemporal dynamics really did behave according to the way the model predicted. So hopefully this sort of brings a new philosophy in terms of using a sort of a synthetic programmable biology to understanding how we can, how developmental patterning occurs and how we can actually engineer it. And so hopefully next time I'll be able to show you an implementation showing patterns, but not yet. So just leaves to acknowledge our collaborators, again, people in University of Cambridge, Jim Hasselhoff's lab in particular, and some colleagues at Microsoft Research. And thanks very much to you for listening.
Thank you, Neil. So that seems an exceptionally rigorous approach to synthetic biology to me, and an interesting target.
A quick question. Did you do a rigorous parameter identification for the model?
Yeah. We used a Bayesian approach to infer the parameters. Basically, we had, I think, something like 25 different experiments that exposed the system to different modes, if you like. And then we compared something like 25 different models as well that had different features, and this was the one that gave the best sort of agreement with the data with as few parameters as possible.
Could you get distributions rather than values?
Yeah. It's I have the full cost series. I think I might have them in extra material.
Yeah. Okay.
Yeah. So we have that, and then similar. That's just for the wild type promoter, but we have equivalent things for the mutant promoters as well. So, for example, we see posteriors like this where the parameter really wants to be 0, so a lot of that values. So all of the probability is heaped up along the left-hand side there. And I should also say that between the 3 different promoters, the wild type and the 2 mutants, they all shared the parameters that describe the signal binding to the receiver proteins, because of course we've not mutated any of that side of things. So the only parameters that were promoter specific were was the actual rate of transcription coming off the promoter, and then the affinity of the regulators for the promoters as well.
Okay, since the tea break's only 20 minutes, I will break this there. Hopefully any further questions in the discussion section from Daniel again.